being so interested in mussels. We've had kind of a interesting, I'll introduce myself first, then I'll introduce the mussels. Um, I'm Wade Motor. I'm the executive director of the Upper Sugar River Watershed Association. We're based out of Mount Horeb. I think as many of you know here, I recognize most of the names here. We do a number of things around the watershed and beyond. One of them being citizen science and the volunteer programs that are housed underneath that, like our dragonfly monitoring program, water quality monitoring, and mussel monitoring, which we're hoping to get kicked off this year after a few years of downtime due to bad weather, flooding, pandemics, all sorts of stuff. So we're hoping to get back on track this year. So as a little background, back in, I believe, is it 2017, Jesse and Lisi?
Yep.
Okay, back in 2017, we got a small grant to try something that I believe is the first time in Wisconsin's history where we've actually transplanted mussels from an estuary along the Mississippi River to the Sugar River. And the Sugar River has a pretty long storied history with mussels dating back to the early 1900s. And in fact, the Sugar River was featured in a New York Times article in the 1920s about the rich mussel population in our river. But that also attracted a lot of people and button manufacturers. So this is before plastics. So there's a period there where a lot of mussels were harvested from the Sugar River and buttons were made from the mussels. So the populations were decimated, the numbers plunged. But despite that, we still have what we think is a good habitat for mussels in the Sugar River. And we're looking to try and track what mussels are out there. We want to track the 300 plain pocketbook mussels that we planted out there back in 2017. So they should be about 4 years older now, and see where they're at, see if they've moved downstream. Any new information we can get from them and that population is going to be really interesting. And we're going to be hoping to get some volunteer help this summer in tracking that. So I want to make some time here for Lisi and Jesse. So Lisi Kitchell and Jesse, is it Weinzinger, Jesse?
Yeah, Weinzinger. Yep.
Weinzinger. All right. You don't have to accept my bad pronunciation. So Lisi and Jesse are the conservation biologists for DNR, and they specialize in the Wisconsin mussel monitoring program. So they go all around the state tracking mussel populations. They're kind of our state experts in mussels. So these are the two people we want to be talking to about this project, and they were part of our project back in 2017 as well. So Lisi, Jesse, which one of you wants to go first tonight?
I think I'm going first.
Okay.
If that's okay. So
yeah, that sounds good.
I'm gonna share my screen.
Very good.
Am I shared?
And
There we are.
Hannah started recording already. Excellent.
And you guys are welcome to, well, I'm going to go through this pretty fast to leave time for questions at the end. So we'll go from there.
You aren't quite sharing your screen yet.
I'm not quite
Not
sharing
quite.
Well, I need to close this first. Wait, I need to get out of here. I had clicked on share screen. Am I now or not?
Not yet.
Okay, let's go back to share screen. Okay, are you seeing that screen?
You're not.
No, you're not.
There it is.
Oh, okay. I have no idea what's going on. Okay, so let me move this up. Let me get back up to where the start is. This first one. And I will get to this. Is that looking good?
Perfect.
Okay, good. Now then. Now I have to figure out how to advance. So anyway, I am Lisi Kitchell, and I'm going to talk about Wisconsin's mussels. I'm going to keep it fast so we have time for questions at the end. One of the first things I'm often asked is what's the difference between a clam and a mussel? They're both bivalves. They have two shells, and you'll hear me use the terms interchangeably. So in vernacular, people call them clams and mussels, and etc. Technically, there are differences. We have our native clams, which is this down here, the pea clams or fingernail clams. These are small. They get no bigger than a dime. And then of course we have an invasive clam, which is this one, which is Corbicula, or the Asiatic clam. Both of which are kind of more round than elongate, which our native mussels and the zebra mussels are. And the Corbicula doesn't get bigger than a quarter. So if you've got something bigger than that, then you have one of our native mussels. Zebra mussels only get to a couple inches as well. So, but you will hear me use those terms interchangeably. So don't be concerned about that. So in Wisconsin, we have 50 species of native mussels. They vary in size from the great big washboards, or the size of dinner plates, down to the little tiny lilliputs, which are 1.5 inches. And they come in all kinds of shapes and size and colors, as you can see. Some have bumps, some have ridges, some are smooth, some have rays. It just depends on the species and what you're looking at. And some are just plain old clams. So mussels occur in lots of diverse habitats, all of our lakes, rivers, and streams. However, the one place where you don't tend to find them is in the headwaters, in the trout waters that are very cold, and they have very little algae for these guys to eat. Plus they have a limited fish diversity, which I'll explain and why that's important. But they're found in, they prefer running waters, but there are quite a number of them that also occur in our lakes and our flowages. Oops, sorry. So what's important about our native mussels is that they're very long lived. They live 10, 20, 30, 40, 60 years. We've aged them at 100 years live in Wisconsin. But what's most important is that they require a vertebrate host, which is usually a fish, to complete their life cycle. They actually need the blood of the fish to develop all their internal organ development. And because of that, they've developed really unique abilities to attract those host species. There's one that actually uses the mud puppy as a host that doesn't use a fish. And I'll go into more detail about these unique abilities. But first, there we go. First, the life cycle of the freshwater mussel. So here we have there are males and there are females, and you can actually, some of the species in the pocketbooks that we put out there in the sugar, you can tell the difference between them by the shell shape. So the males release the sperm, gets taken in by the females, it passes through her gills, which have all the eggs along them. They're called marsupial gills. That fertilizes all those eggs. Those fertilized eggs develop into what's called a Glucidia. And this Glucidia is a snappy little Pac-Man kind of thing. Its sole purpose in life is to get onto the gills of a fish and to get that fish blood. If it does not do that, that's it. It's not a free living kind of organism. But if it gets onto the gills of the fish, it then in the presence of that fish blood, it develops all of its internal organs, and it develops into what is a juvenile mussel. And that juvenile mussel drops off and then can continue on the cycle of life. So bottom left, you see these dots. This is a fish gill, and these dots are those Glucidia in the fish gills. Over here on the right-hand side is the mud puppy, which the salamander mussel uses for its host. So because it's so important to get onto the gills of the fish, they have developed lots of unique ways. And of course, one of the best ways is food, fish-like food. Some of these guys have developed, they pack all those Glucidia into an egg mass. In this case, it looks like a worm, and you can see the fish looking at it. Here is these pink things. I don't know what they're supposed to look like, but the fish eat them. And down here are more from different mussels, and these are thought to look like scuds. But there's lots of different variability in terms of what they have developed into. So this is one that releases, this is a female shell here, female releasing. She's releasing those egg masses called conglutinates, which here they look like, if you look at them, and if you looked at bugs on the bottom of rivers, they are not just an egg mass, they have eye spots and a head capsule. They're trying to look like a bug on the bottom of the river, and they're sticky on the bottom here. So when she releases them, they stick to rocks and things like that. And then the fish come along and they eat what they think is a bug on the bottom of the river. And then that's how the the Glucidia get onto the gills. So the Glucidia are very light and small here. These dots here are the Glucidia from that egg mass. And you can see they're very light. So when the fish gape and sucks, which is how they eat, these guys go along with the water that goes over the gills. That's how they get onto the gills. So some of them have really specialized. So this is a picture of a black fly larvae from my biological text. And then these are those conglutinal masses from a mussel that are trying to look like black fly larvae, because that's what their host fish eats. So it's got eye spots, it's got a head capsule looking thing, it's got an antenna looking. And so that's what it spits out. With the hope of getting the fish to eat that and then get onto the gills of the fish. So how a clam or mussel knows what a black fly larvae looks like, I have no idea, because they do not have a brain, nor do they have eyes, but they have specialized in that fish host. So if your host is a predatory fish, you're not necessarily going to want to, I mean, you could spend all your time looking like bugs, but the fish are going to go, if a predatory fish is going to go for another fish. So this is a mussel that has developed an amazing fish lure. So the soft parts of her anatomy, her mantle, has developed this lure. So this is part of that mussel that's here. So she puts this lure out to attract that predatory fish. You can tell these guys are not interested because they don't eat other fish. But this is, to me, is an amazing lure. It has an eye spot. It has, you know, things that look like fins. This is just the soft parts of her shell, or not her shell, of her mantle that has developed into her lure. And what happens is the predatory fish comes down to eat the minnow or whatever it thinks it is. And when they do that, she pulls the lure in really fast, and then she spits those Glucidia on into the mouth of the fish, which then again go into the water that's going over the gills and get onto the gills of the fish. So the intent here, she doesn't put it in an egg mass, she just puts it into the mouth of the fish when the fish comes in. It's very rare that you see these fish lures are at all, that they're removed or tagged or anything. She is so fast at pulling it in and so fast at spitting it out. So here are some other fish lures that are not quite as fancy dancy as the other one, but these are other mussels. And here you can really see that it's part of her soft tissues. It's part of her mantle that makes this fish lure. And here's another one. Again, different species have different looking lures because that's what, that's what they do. But not all fish eat just other fish. Some eat crayfish too. So this is one that has a crayfish lure. So here's the shell, the outer shell of the mussel, and it's starting to put out its lure. And here its lure is completely out. It looks like a crayfish. It's got these things that like legs and this back part that it flips. And I forgot to say that with the other mussels. They flip the fish too to look like a fish. And so she flips this crayfish. It looks like a crayfish dashing under a rock. And again, the predatory host comes down. These white globules are those marsupial gills that she spits the Glucidia out of. And this is a small little mussel. It's called the rainbow. And it spits that into the mouth of the fish. And again, that gets across the gills and get those Glucidia latch onto all those gills. And we have the next generation started. So not all of the mussels take even chances of that. This is a mussel that actually grabs a hold of the head of the fish. This is the snuff box. Again, it's a small mussel. It puts out this, it comes up and it has this thing that might look like a lure, I'm not sure, but it's very iridescent. So the fish are attracted to that. It's fish that are darters, that its host fish are. And it specializes in primarily these certain kinds of darters, and they poke along the bottom. They see this, they poke into there, and she snaps a hold of their head. And so the poor fish, you know, picks up, he's got a clam stuck on his head, he's flailing in the water, but you can see the fish's gills flaring as the as she pumps her Glucidia onto the gills of the fish. When she gets done, she drops off, and the poor fish shakes his head and swims away and goes, "What just happened to me?" But that's how they ensure it's that it gets on the right host. So some species use a variety of fish, such as they might use catfish, or they might use minnows. This one specializes in log perch as its host fish, which is a kind of darter. And then they don't all have fancy dancy things that they do. This is our rarest mussel, which is the winged maple leaf. And we looked for a long time trying to find the host for it because it's critical. And that one of the best ways to look for the host is to see what the mussel does and then say, okay, then who would be the appropriate host to do that? Well, when these females come up, they kind of do this thing. They just kind of exude this thing they call an eruption. And when she's ready to, when the Glucidia are ready to latch on, she kind of just pukes up a big gray gob on the bottom of the river. And within minutes, there's a catfish on it. So probably there's something olfactory about that, but you know, it doesn't need to do anything fancy dancy for a catfish because catfish are happy with gray gobs on the bottom of the river. So again, it comes back to all these types of mussels, you do a technique to attract their host that is specific to that host, and some are very specific. These guys only use channel cats, but some of them use a whole spectrum of hosts. Oops, I keep hitting the wrong button. So why care about mussels? What good are they? They have been used for centuries as food, tools, and decoration by the Native American Indians. People often ask about eating them. I tell people that I wouldn't these days because of contaminants. They're really good at concentrating contaminants. And the Native Americans didn't have that. You'll find piles and piles of shells from their fishing grounds when they would go out and collect these guys. And Wade mentioned earlier, on the Sugar River was known for its shell material. There was a pearl rush at the 1800s that there were places advertised as far as Paris, France, that the Sugar River was the capital of the pearl industry. And what happened was just wanton waste. People would wade into the river and just rip open the shells looking for pearls. And certainly there were people that made their millions. I guess it was a hotel in Paoli that was paid for entirely from the money he made off the pearls in that pearl rush period. And then the next time they became important was in the 1880s to the 1940s for the pearl button industry. And once again, the sugar contributed lots of material for these buttons as they were cut out as blanks. All of these piles are piles of shell material that has holes cut in them. That worked. The pearl buttons went well until the 1940s when plastic came along and kind of replaced that whole pearl button industry. The next time they became important was in the 1960s. The Japanese were culturing pearls, and they went around the world looking for the best cultured pearl, and lo and behold, or the best nuclei for a cultured pearl. The upper Mississippi was the best, and it's simply a function of our soil. We have high calcium carbonate soils, we have hard water, but it makes really good shell material. So they came and bought lots of shells and took them back to Japan. And that went well until the zebra mussels invaded, and then that kind of closed that cultured pearl industry shell material. So how important are mussels? Biologically, they are our natural filters. They concentrate contaminants that are in the water column. They clean up our rivers. They're very important to water quality and water clarity. They provide fish or food for fish and wildlife. They provide habitat the shell material, as well as food for benthos, which is the stuff that lives on the bottom, such as the insects and the crayfish. They concentrate all that stuff from the water column, and then they spit that out, and that's what those guys eat. They remove pollutants, as I said. They're great biomonitors. If you start losing the mussels, you obviously have some kind of water quality problem. And they're important to the stream processes. They move around in the bottoms of the streams, not unlike what earthworms do in our soils. So they keep things from being super embedded in the bottom of the rivers. So here's an example of them as natural filters. Both tanks started out the same. Put one clam in here, 1 hour later, and it had cleaned this water all up. They filtered tens of gallons of water a day. And if you have a mussel bed, you've got hundreds and hundreds of gallons of water that are being filtered. So they're really important to our stream water quality. But we still have mussels that are at risk. 27 of our 50 mussel species in the state are endangered, threatened, or of special concern. And there's others we're keeping a watch on. So what are things that cause this? Dams, impoundments, alter these running waters, which is what a lot of these species evolved in, to a series of lakes. Now, some of the species do fine in lakes, and some of their fish hosts do fine in lakes. But if they don't have both, these impoundments have precluded a lot of populations of mussels, and they also serve as barriers for those mussels. We still have sedimentation coming in. We found out that our mussels are highly sensitive. The juvenile mussels are highly sensitive to ammonia, nitrates, and nitrites. So often we'll find mussel beds with just older guys in there and not any young, because the young are not able to survive in the presence of some of the sedimentation, as well as urban runoff. So all of these things are contributing to water quality. And if you're killing off the juveniles, then you don't have future populations. And of course, invasive species, the zebra mussels, which have a high affinity. There is a clam in each one of these piles of zebra mussels. They have a high affinity for our native mussels. So that's been a major problem. But there's hope. So the Genoa National Fish Hatchery has been propagating mussels for us to try and replace some of these lost populations and reintroduce them to areas that for whatever reason they have not survived. And Jesse will talk more about that. This is where our pocketbooks came from in the sugar. So what can you do to learn more about mussels? You can request a Mussels of the Upper Mississippi River booklet, or you can go to our mussel monitoring website and download that. The Mussels of the Upper Mississippi covers pretty much all the species in Wisconsin except for a couple, or just Google. Here is our actual website link, but you can always just Google Wisconsin Mussel Program and it'll usually be the first thing that comes up. So what else can you do? Just go out and collect mussels. They don't run fast. You don't have to kill them to collect the shells. So they're a pretty easy organism to go out and find. And when you do find them, pick up shells, take pictures, report what you find. You can report them on iNaturalist, which Jesse will talk more about later, or send pictures by email to us, myself, Lisa Kitchell, at this at wisconsin.gov, or Jesse at wisconsin.gov. Sorry, I see a typo there. Anyway, so you can report these guys, and then you don't have to be able to identify them. You can report them. We can identify them and tell you what they are. But if you want to take a chance at identifying them, my experience has been people within a year or two, they're good to go at identifying these guys. So if you want to, I'm going to make sure I'm not going too far. So if you want to look and see what's in your own backyard, we have observations by county on our website. You first have to pick a county. Oops. And the reason we do this is there's only 1,001 sand creeks, mud creeks, leach lakes, etc. You pick one of these water bodies, if that's in your backyard, or if you're just interested in that water body, and then it will give you a list of what species have been collected from that creek. Now, there are many, many water bodies that have no collection records. And so then you may not have that, and that would be a good place to go out and look for mussels. So that's it. That's where you can find information about what's out there now, but these are by no means comprehensive lists of collections. And so any and all things, we'd also love it if you could update. These are fairly current, but we have some records that go back to the 1970s. So by all means, get out there and join our mussel monitoring program. Okay, I'll leave it over to Jesse now.
All right. Thank you, Lisi. Did we want to see if there were any questions first?
Sure, I was just going to wait to the end, but this is fine if people have questions now.
Otherwise, I'll try and get my PowerPoint ready to go.
Yeah, if anybody had any questions, go ahead and just unmute and ask.
Looks like there might be one in the chat from Maggie. She's assuming that when you say collect mussels, that means to photograph them and put them back.
Yes, if they are live. And the other thing is, if you don't know which end is up, just lay them on their side. And the reason I say that is if you put them upside down, it's kind of like the turtle on his back. They have to like wiggle, 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 well, to get the right end up. So it's easier just lay them on their side and they'll burrow themselves back in. Otherwise, you can collect the shell material. I highly recommend rinsing it off before you put it in a Ziploc and seal it up. Otherwise, that river mud will get really stinky.
All right. Can you guys see my screen okay?
Yep.
Fantastic. Okay. My name is Jesse Weinzinger. Wade and Lisi already introduced me. As mentioned, I am a conservation biologist for the Wisconsin DNR. I'm stationed in Wisconsin Rapids. So basically the dead center of the state. Lived in Madison for 1 year. And actually at Canoecopia, I was talking to a volunteer from the Upper Sugar Watershed, and that's kind of how this plain pocketbook project started. So it went just from a casual conversation to actual really interesting CBM project. So I'll share some of the information from that project and what we're going to do in the future. But before we do, I'll just provide a general overview of the Mussel program, kind of some of the things we're doing here in the state, and then we'll go into a little more specifics about some of our volunteer opportunities in the sugar. So as Lisi mentioned, that Wisconsin is home to 50 species, 24 of which are state endangered, threatened, or a special concern. That number used to be 52 species. However, in the last 10 years, we have deemed 2 of the species extinct because they have not been documented in 50 years. That is the scale shell and the fat pocketbook. There are 2 additional species that have not been documented since the 1980s, the ebony shell and the elephant ear. So that number, may in the near future dramatically decline down to 48 plus or minus. And that's just not an occurrence here in the state of Wisconsin, but that's worldwide. Freshwater mussels are regularly considered the most imperiled animal group in the world. Some of the causes Lisi already mentioned. You have several water quality issues. Down in the southern half of the state, you have a lot of non-point pollutants, whether that's nitrates, ammonia, even phosphorus can indirectly contribute to mussel declines. Up here in central Wisconsin, even northern Wisconsin, 100 years ago, you had big logging industry. So just floating the logs down the river, you would actually scour out riverbeds, displacing and killing mussels, but you'd also increase or decrease the pH. And because mussels build shell because of, from calcium carbonate, they need higher pH concentrations. So when you have more acidic waters, that will decrease the thickness of their shell. Also, you had a lot of pollutants from paper mill industry or a lot of industry in general. So it wasn't until the Clean Water Act where a lot of those pollutants were very common throughout our watersheds. More recently, some of those pollutants have gotten cleaned up. So we're actually seeing a rebound in some watersheds in the state for various mussel species. Connectivity barriers. Now, that basically a fancy way of saying dams, but dikes and other water control structures can also be influenced by that too. But dams are the big one because Lisi mentioned that most mussel species rely on a host fish. So if the host fish can't travel upstream because of a dam, the mussel's not going to be able to reproduce and complete its life cycle. So the two mussels I mentioned that haven't been documented since the 1980s, their host fish is the skipjack herring. So that the skipjack herring would actually migrate up from the Gulf of Mexico all the way up into the headwaters of the Mississippi Basin, whether that be in Montana, whether that be in Minnesota, whether that be in Wisconsin, and the mussel would be able to reproduce. But when the lock and dam system started and during the New Deal in the 1930s, the skipjack herring was more constricted and restricted from going upstream. So below the lock and dam system in the Mississippi River, those species are still very prevalent. But as you move upstream, up into the basin, they become very rare and borderline extinct in some of the northern states. Climate change, we could talk the entire time about that issue. It's not just temperatures, but it's the hydrology itself. You think of greater droughts, which I think Maggie is from Texas. There have been a lot of drought conditions that have helped lead some of their mussel species to go be listed as federally endangered in recent years. But contrary to that, there are massive floods. And here in the Sugar River is a prime example. You had, two 100-year or two 1,000-year floods almost back-to-back years. And that has actually changed the entire habitat in some sections of the Sugar River. And then invasive species, as Lisi showed, they can encapsulate native mussel species and either suffocate them or displace them. So we see that in the Great Lakes and some of the Winnebago system, tributaries and lakes, in the Mississippi River as well. So just to the right here, as mentioned, one of the major pollutants in a lot of the state is ammonia. So mussels were not considered, or they were not tested for ammonia concentrations by the EPA. So when they wanted to test or implement standards for ammonia, they did not take mussels into effect. Well, studies started coming out that showed mussels were very sensitive to ammonia concentrations. So when they started testing freshwater mussels, they realized out of all species of all taxa groups, whether it be trout, whether it be mayflies, whether it be other invertebrates, 8 out of the 10 most sensitive animals ever tested to ammonia concentrations were freshwater mussels. And that's just one example. But if you expand on that, chlorides, from road salts, de-icing practices, very commonly used throughout the state. Mussels are among the most sensitive organisms to road salts as well. So compound that with several other contaminants, and as mentioned, freshwater mussels are regularly considered the most imperiled animal group in the world. This graph here shows worldwide imperilment rates for various animal groups. And there's a lot of focus on, protecting birds and mammals, but relatively speaking, freshwater mussels are much more imperiled. And in part of that, not only of their sensitivities, but when your host fish is a freshwater fish or an amphibian, and over a third of their populations are in decline, the mussels aren't going to be able to reproduce. So it's just a compounding factor. And so we regularly like to consider mussels as the kind of the gold standard in conservation. So the Wisconsin DNR in 2009 established the Wisconsin Mussel Monitoring Program. And it was just to provide the foundation for various programs, whether it be volunteer groups, schools, nature centers, to implement conservation practices regarding freshwater mussels. So we partner with various groups to lead field trips and training opportunities. The Natural Resource Foundation, we partner with them in a non-COVID year to provide various field trips throughout the state. We conduct salvages. So whenever there's a dam drying down the reservoir, that usually, exposes mussels to the air, and therefore they can't breathe. And we have volunteers go out and just collect, document what's there, and then throw them out into the river out of harm's way. Mussel Blitz in 2014, there was a new species discovered. The species is not invasive. It's just newly documented in Wisconsin. We have some occurrences in the UP, Ohio, and the Eastern Seaboard, but never in Wisconsin. So when it was first documented in the Eagle River chain of lakes, we wanted to see how widespread it was in that 28 lake system. So that was in 2018. We did a mussel blitz to document that. The reintroduction and augmentation of the plain pocketbook happening here in the sugar. And then we partner with various groups to do watershed-wide assessments. And this example here is just with the Milwaukee River. So what do we do with those data? Sometimes there are volunteer programs where, you lead a training and then you go out and then you submit your data and then you don't know what happens to it. So we tried to really be as transparent as possible, allowing that our volunteers to see what we're doing with our data. So we have our annual mussel newsletter, which is going through editing right now and should be available here in the next month. And we have our online GIS platform that shows where mussels occur throughout the state. But whenever you provide a mussel observation, it will directly get imported into our DNR, our General Mussel Atlas, the DNR database. And those occurrences, say if you came across a threatened or endangered species, resource managers will have to take that in effect when they do resource reviews. So if they want to build a new bridge or, provide new riprap on their bank, they'll have to take that in account if there is a listed species in the area. Federal governments will use, every 5 years, they do species status assessments. So they want to know where the most recent observations are for some of our rare species, and volunteers have contributed to that. And then, as mentioned, we share with volunteers through our newsletters and GIS platform. So to the right here, I don't know if you can see it very well, but it's a map of the state. There are 3 different dots on there. The gray dots are a little smaller, and that's just showing you where there's a mussel occurrence in our database. So some of the river systems like the Chippewa watershed, you see lines going all the way up, to its headwaters near Vilas County. And so you can almost make out the entire river of the Chippewa. But others like the midsections of the Wisconsin, the Sugar, not so much, and various other river systems, we have very little information. The green dots show where DNR surveys have been conducted since 2015. DNR or partner contractors have done surveys, but the blue dots show various CBM or volunteer observations. So you have a lot of new species and lake occurrences in northern Wisconsin in the Big Lakes region. The Milwaukee Watershed has provided a lot of new information for mussel occurrence, but it's just helping us fill in data gaps that we otherwise may not get to. So Lisi mentioned the website, and we'll see if we can just briefly go into that. Can everybody see the website? Or is it just the slide right now?
It's just the slide.
Oh, okay. Well, maybe we won't get into it. But we have various resources. If you just type in Wisconsin Mussel Monitoring Program, we have various resources that will show all these. You can download the digital field guide. You can collect this field guide from the Chicago Wilderness, which gives you great detail into the specifics of what are the key identifiers for any mussel. And it's a free download. That's really a key resource I used when I started learning about mussels. And then our website has that GIS platform. So if you go into the About Mussels tab in the upper left there, there'll be observations by county. And then you can select Dane County and the Sugar River, and you can see about a half a dozen species that have been recorded in the last 30 years from the Sugar River. So instead of trying to learn 50 species, you can start at those 6 or 10 species that occur in your local watershed. Otherwise, there's some other books. There's an app called Clam Counter or the Canadian Freshwater Mussel Guide. And the other app, which I'm going to briefly go into, is called iNaturalist. And it's a great open source project developed by National Geographic and UC Berkeley, made up of over 1.5 million users that have contributed over 65 million observations worldwide. Everything from fungi to bats to mussels to plants. It's a naturalist one-stop shop, basically. And all you got to do is create a username. If you're interested in mussels, join the Freshwater Wisconsin Mussel Monitoring Program project page, and then start submitting observations. And here's the 1, 2, 3, record your observations, share with fellow naturalists, and discuss your findings. So what it looks like on a smartphone is you open up the app and it provides the gallery of your most recent observations. Now this is from a Google Pixel, but it looks very similar on an iPhone or Android. So all you do, you see your gallery of recent observations, and say if you're canoeing and you come across the shell, all you do is hit this little add button here. Either take a photo directly or you pull it from your gallery in your photo app, and it automatically geotags your location if your location services are on. It records the time and date, and it will even identify what's in your hand. So here you'll see that we're pretty sure it's in the subfamily Emblemeidae. Well, that's the correct genus for what the species is, and their top suggestions are 3-ridge, and that's actually correct. Now, it's not always correct because some mussels are coated in algae, but it gives you some guidance as to where to start. And then you just submit that. You know the water body name where you found it, the county, and there's optional information, but those are the only 3 information you need to submit. You hit submit and you're done. You can provide an observation without cell service. You can take a photo in the field and then log it when you get back home if you want. It's very, very intuitive. I don't think I've ever had a glitch on it in the 3 or 4 years I've used it. So iNaturalist app, consider looking into it. Otherwise, there's also a website if you wanted to explore it further. But outside of that, we have 2 monitoring strategies. We have casual observation and the time search. Now, the casual observation is if you're out on the lake one day, canoeing, paddling, or you're at the beach and you come across the shell. Well, that information is very useful because it might be an endangered species or it might be a common species that we may be, the more information we have, we may see declines or increases in their occurrence throughout the state. So we will take everything from a giant floater, which is arguably the most common species in the state, to the winged maple leaf, which Lisi mentioned is our rarest mussel. If you wanted to go in depth, provide a little more semi-quantitative stuff that would provide us abundance data, species richness, we have time searches, which those are just 15-minute intervals adding up to 2 hours at a given location. You can use mask and snorkel, or you can use bathoscopes. And basically you're just fanning the substrate for 15 minutes at a time. At the end, you gather all your shells, take a photo if you don't know what you're technically looking at, submit it to iNaturalist, or email us the photo of all your shells and we can identify it for you. How to monitor for mussels. We have in-person trainings that kind of gives you the foundation of where to build from. And then we also have survey and monitoring outings where we actually go out and do those time searches. Sampling locations, you have shallow water areas. Generally you want to target in certainly wadeable areas, unless you're an experienced diver, then you can go a little deeper. Stable flows are really key to providing good habitat for mussels. So if you just see cobble and boulder on the bottom or bedrock, or if you just see straight sand that's really loose, you're not going to find many mussels in there. So you look at areas like on this photo where you got a good mixture of sand, gravel, and maybe even a little bit of cobble. It's not a race, so go slow. As mentioned, fan the substrate to expose several buried mussels. It's very common that they're completely buried. And just develop a blue mind, which is get your focus kind of your focus becomes on the mussel and you being in the water instead of thinking of the work you have to do back home or the cleaning you have to do or anything like that. There's actually a great book about it. And just to touch on the mussel reintroduction, Wade already did a great job. But back in 2017, thanks in part to Bill Keene, he was wondering, well, the sugar, I don't see mussels in there. Where are the mussels, basically? And I was like, well, there could be a lot of water quality issues, but I could go check to see if we have any records from the upper sugar. And sure enough, we did. And it wasn't 2 weeks later, we had a regional mussel meeting near La Crosse, and the National Fish Hatchery said, oh, we have a whole bunch of extra plain pocketbooks. And I was like, oh, well, there's plain pocketbook in the sugar. And then I got back to Bill and Wade, and we developed a CBM grant to augment their current population. So we met up in Paoli a year later. So some of these little mussels would get grown out. And we actually did a little mussel workshop to place them. We measured all the 300 individuals, so we have age length data. As you can see, they're about 1 inch long, and these guys can get out to be 6, 7, 8 inches in diameter. And then we'd move them from the cooler to the volunteers, and they'd actually plant them in. So we gave them 2 years to come back and to measure their growth and look at their survivorship. And this was a year after a massive flood occurred in Paoli. And we couldn't just find the mussels, but the entire substrate of the river shifted. So what used to be a great fine sand gravel mixture became gravel cobble. So the whole bed load got shifted downstream. And so this year, now that COVID is starting to taper off, and we're allowed to do CBM events again, we were partnering with the Sugar to hold a couple workshops, hopefully identify where these mussels have gone, and again, measure their growth and look at their survivorship. But in addition to that, we'd love to know more about the distribution throughout the entire watershed itself. And so without going into detail, these are some of the species that are common in the watershed or were common. As mentioned, the plain pocketbook is a very large individual, but you can see spike and fluted shell. And then there's a couple state listed species, the lips, the elk toe, and my favorite, the pistol grip. If you look at it, you can kind of see a resemblance of an old school, Magnum pistol, or another common name is the buckhorn. And you can kind of look at the knobs at the base of an antler and see those nodules on the shell. So that's all I have, but we look forward to meeting you in person in the near future and going to look for mussels.
Great. Well, thank you, Jesse and Lacey. Do we have any questions from the group? Doesn't look like the chat's been too busy, but if anyone has any questions, please unmute yourself and we can quiz and question Jesse and Lacey. One question I had, if everyone's too shy, from Lacey's presentation about the reproduction cycle of mussels is so fascinating, and all the different moving parts that have to happen just to create more mussels is pretty incredible. I was wondering if there is any research or reasoning how when the male releases the sperm initially, is there a way that the females collect the sperm, or how does it seems like it just kind of goes into the river? Is there a rhyme or reason to how that takes place?
Yeah, there actually is. I was just trying to keep my talk short and sweet. I could talk for hours. So we found we used to think that they just released it into the water column and hoped it got the right female. Instead, we found out that they released the sperm in what are called sperm balls, and the sperm balls are the head of the sperm are all like together with all their little flagellate tails sticking out, and it looks like some kind of little I shouldn't say quality, but some like little water animal. And that sperm ball actually goes out into the water column and actually goes into the right female. So if you put a fish tank and you put a male of one species at one end and you have 3 different species of females, when it releases that sperm ball, it goes and goes plunk and goes right into the right female. So there's got to be some chemistry there that it knows how to find the right female, but that's how it does. And then once it enters that female siphon, because that's how they take in everything from the water column, then it just breaks apart and it fertilizes all those eggs that are along the gills. So that's how that magic piece occurs. So, and one other thing I wanted to bring up, we talked about aging mussels. If you look at, this is a good slide to look at, what happens is the mussels in the wintertime go down and they quit growing. And much like our trees, they quit growing in the winter and then they start growing in the spring again. And that's what these guys do. So if you look on the shells, you'll see lines. Yeah, Jesse's showing you lines. And those lines are annulized. So that means that those mussels at that time went down. In the spring they came up and started. Some mussels grow faster than others. The giant floater to the right grows, you know, much more rapidly. It only has 1, 2, 3, 4, maybe 5 growth rings. And that's how we age them. Different species grow at different rates. And the problem is for the older ones is the shells erode at that oldest part of the shell. So it's sometimes hard to find the first couple quote unquote annuli. And when they get really, really old, the annuli get really, really close together and they're very hard to tell apart. But that's how we age them, just like you do tree rings.
Yeah, and you can see, as Lisa mentioned, some species grow really fast. So the giant floater is a great example, but so is the plain pocketbook. You can see a growth line here, growth line here, growth line here. And so this individual is probably about 4+ years old. So when we find these animals in the sugar, these animals could be 4, 5, 6 inches in diameter by now.
Yeah. And they could be reproductive. So a lot of times they'll grow fast until they get reproductive and then they'll slow down their growth because they're not putting it into shell anymore. They're putting it into reproduction.
That's incredible. Mussels are pretty incredible. Any other questions people may have? I have plenty more, but.
Have you noticed with any kind of acidification issues with over time from older shells that have been collected in the past to now? Has there been any thinning of the shells that you've noticed?
Not really. We have some river systems that have thinner shells because they're more tannin, they have more acid in them. But for the most part, a lot of Wisconsin is hard, quote unquote, hard water. So our shells are pretty thick. You go to other parts of the country, New England, for instance, they have very soft water. And I have picked up shells and put my thumb through the shell because they are so thin, just because they don't have a lot of calcium carbonate in their waters. And that's true on the Pacific coast as well. We're really in the heartbeat of good mussel quality, good dense shell material, but haven't seen the changes over time in terms of the thickness of the shell.
Mm-hmm. Yeah, we haven't done studies on that, but they're out there. Some of the big museums like Ohio State, they've actually looked at specimens from 100 years ago and compared them to today. So that information is out there.
Thank you.
Good question. Anyone else with some questions for Jesse and Lisi?
Just a quick note about the sugar. It really suffered from that pearl rush period and then the pearl button period. So it used to be thick with mussels, and those mussels unfortunately declined with that over harvest. And you know, it's still kind of coming back. So let's keep working on getting it to come back, not giving it poor water quality to try and come back in.
Yeah. So even an observation of a shell that you find on a stream bank, you know, you don't have to do these time searches, but if you were just haphazardly to come across the shell that the local muskrat threw on the bank, we would love that information, because it might be the first observation in the for the Sugar River.
That's a
And
good point. I remember you saying something about even, finding dead mussel shells with no mussel inside, just the shells, are still a good observation to make.
Yeah,
Yeah,
because that assumes that that mussel came from that river. And so if you find the shell, if it's weathered and bleached out, that assumes that's an older individual. But that also means at some point that that mussel was alive in that system. So yeah, it usually associates the presence of a population.
I have a question. You, in the training, or you had written down, July 29th and July 31st. Were those in-person events?
Yep. We've been talking over email with Wade about these. And so July 29th, that in-person event would be at Paoli. Is it confirmed at the place we've had it in the past?
Correct. Yep. At the mill. Yeah, the mill in Paoli.
Okay. Sorry about the pronunciation. And then the 31st would be another event downstream. The location is yet to be determined, but we're probably going to loop in the Lower Sugar River Association to hold an event somewhere south.
So those events haven't been put on our website yet, but we're just kind of collecting details still on that. So they'll be on our website very soon, and we'll send out an email when they are ready. And I think so there's another question in the chat that kind of alludes to it. Is there an in-person training to go with this training? And in the meantime, is iNaturalist the way to be involved to report sightings? So yes, I think the answer is yes, that our in-person trainings are being scheduled, and they will be, so like the 29th and the 31st are both going to be, I think, identical trainings. They're just going to be in different locations. So if you can only make one, that's fine. You don't have to make both, but maybe Jesse or Lacey could maybe talk about what the plan is on those workshop days.
Yeah. Do you want me to go, Lacey?
yeah, go for it.
Okay. Yeah. So let's say it's a similar group to this, or you guys show up and maybe a couple others. We'd really focus that training on how to actually, first, how to identify mussels. We'd go over the identification of, those half a dozen to 10 species that have historically occurred or we have recent observations of, including the plain pocketbook. So that would probably take about half an hour to an hour, depending on how in depth we want to get. But then we go over the monitoring methods and actually get in the river and do a survey. So it would probably be about a 2 to 3 hour training total, but really kind of focusing on identification and monitoring methods.
Great, thank
Yep.
you.
Yeah, it's just an opportunity to get in the water and actually get experience looking for mussels and finding them. So
Yeah, and if we do find that population in plain pocketbooks, and we're gonna try and look for them in June. If we find them, that July 29th workshop might turn into, okay, let's actually search for those plain pocketbooks, gather which ones we can find, and then do the age length measurements on them and compare it to what we were finding in 2017.
yep, and
Right.
they had black spots put on them. We didn't tag individuals with an individual numeric tag, so we knew each one was what. But they did have black spots put on them, so that's a good way to find the ones that we put in, if those indeed those black spots stayed on. So hopefully we'll find them. Unfortunately, the year after we had this horrendous flood. So we're thinking they may have been washed downstream.
Yeah, Wade, didn't you say a trailer got taken from the local bridge during that flood? The water
No.
was so high.
Did I say that? I
Somebody
don't remember.
somebody was mentioning the water was over the road in Paoli.
Oh yeah, it was. Yeah, it was. I think it reached the bridge at one point, so like it got that high. So it was just a historic flood.
Yeah.
I mean, unbelievable. Does anyone have any other questions? This has been a really good talk. I always enjoy hearing Lisa and Jesse talk about mussels because they're just so interesting, and I think they're kind of a species that gets overlooked, and they do so much for our water resources and are just so, they have such a special history in Wisconsin. So it's a really cool way to involve volunteers and get another feather in our citizen science monitoring cap as an organization. So I hope we can continue this program, and I hope you can join us in either July 29th or July 31st. We'll get those on our website soon once we get some details going for that. Any last minute questions before we close things up?
I was just going to say it's easy to go out and wade for them. You don't have to have a mast and snorkel or a bathoscope or anything. You can just walk along. Also, you'll find piles of shells along the stream bank sometimes from, I didn't go into this before, but in middens from muskrats or otters or raccoons. That's a great way to find lots of different shells. And it's just easy to go out and look for them. Like I said, they don't run away fast. So you can collect them and document them, and you can just save the shells and look at those later. So they're really easy to work with.
Mm-hmm. And I'll just reiterate the iNaturalist app. It's not just for mussels. So you'll see, when I took a screenshot, whenever I did this, my latest observation was for a terrestrial invasive species that was overtaking my my in-laws' burn pit, and I didn't know what it was, so I took a photo and learned that it was bittersweet nightshade. So even if you have any interest in learning what plants or animals you have in your backyard, it's a really useful app for really anything outside.
Yeah, I'll second that. It's a great app. I use it all the time when I go on hikes and stuff. So really useful to have. All right. Well, thank you, Lisa Kitchell, Jesse
There's
White.
one more question in the chat. If I
Oh, I'm
could-
sorry. Yeah, go ahead.
That's okay. It's from Maggie. Are there some mussel shells that are illegal to keep?
Technically, endangered species are, you should have a permit to have them. Having discussed that with our law enforcement folks, they said, well, I'd be bringing you these shells to have you tell me if they're endangered or not. Anyway, and I personally believe that if it's a dead shell, don't go out killing things. But if it's a dead shell anyway, that there can be more information gained by having picked that up and looked at that shell and learned what an endangered species looks like, than say leaving it in the lake, river, or stream. And the wardens have said, well, we're probably not going to enforce it if you have some dead shells. If they obviously were alive and you killed them, which you can tell by the condition of the shell, then that's a different story. And that is an issue we had with some poaching that was going on, but it appears that has ended. The one other place that they should not be collected from is in the St. Croix, the upper St. Croix and Namakagon rivers, because that's under Park Service authority, and you're not allowed to collect anything from there. So if you're happen to be in those water bodies, I would say don't. But if you see an old, dead, whatever in the Sugar River, I would not be concerned about you having that if it's a listed species. And as I've talked with the wardens about it and said, you know, I'm going to tell people they can keep these shells. And they said, that's fine as long as they don't sell them or put them on the market or do something like that, that would then cross those lines, which it makes perfectly good sense. So if you're just keeping them for your own collection, and you want to know also all the listings that are put on iNaturalist, Jesse goes and verifies those. So you know kind of what you have, not kind of, you know what you have. So I wouldn't worry about that. I think you can gain more knowledge by having that in hand than you do by leaving it in the river because you think it might be endangered, because it might not be an endangered species. So I think I'm giving you the okay on that. And if somebody asks you, you can say, I gave you that permission. So
Great.
it's a good question to ask.
Okay, thanks, Lacey.
You bet.
I will make one more plug before we leave tonight. So 2 days from now and Thursday, we have a training session for our dragonfly monitoring program that you can sign up for on our website, uppersugar.org/events. You can sign up on our website for that training. It's kind of similar to this, where there's going to be a nice question and answer session along with some cool facts. We have some training books. We have we have a array of things to deploy volunteers all around the watershed to monitor for dragonflies. So if you're interested in that program as well, that's this Thursday. Just check out our website and sign up there. If there aren't any other questions, I will say thanks again to Jesse and Lacey. Can't wait to see you guys again in July for our in-person trainings and get in the water, and hopefully they'll be warmer. Hopefully the water will be warmer and lower and not flooded and all that good stuff. So thanks again to you guys, and virtual clap to all of you for attending and being part of this. So take care, and we'll hope to see you on another virtual event coming soon.
Great. And thank you all for your interest in mussels.
Yeah, and thanks for having us.
Thanks, you guys.